hunting for fossils at a roadside quarry in mid-Wales. Higher up the slope, Dr. Diane Edwards, a paleobotanist from University College, Cardiff. With her assistant, she's looking for plant fossils in this outcrop of Silurian silkstone, just over 400 million years old. Here, some very interesting and important finds have been made. But plant fossils are often much more difficult to find than animal remains. The collected material is carefully wrapped up for removal back to the Department of Plant Science in Cardiff. One very early fossil plant discovered at that quarry may be one of the first vascular land plants. Let's see how the paleobotanist finds out about this. The fossil must be revealed as fully as possible by carefully scraping away the rock from round it, using a tungsten needle. It's very tiny. When this process of dégagement has been completed, if we pour a liquid of the right refractive index over the rock sample, this is 50% meths, we can see the fossil more clearly. This plant is called Cooksonia, and it's been discovered that it is a vascular plant. It's got sporangia, spore-containing organs, and other interesting features which are associated with life on land. One technique for examining the structure of such a fossil specimen is the film pull technique. The solvent is evaporated to leave a thin cellophane film over the fossil. When this is carefully pulled off, it brings with it a thin layer of the organic material from the plant. If this is mounted and stained on a microscope slide, we can see some of the actual internal structure of this 410-million-year-old plant. We see tracheids like these. They're water-conducting cells, thin tubes, stretching from side to side across the picture. The little cross lines are strengthening material in the cell walls, and they're clearer like this on a scanning electron microscope picture. The presence of internal conducting and supporting tissues, such as this, shows that Cooksonia was a land plant. Earlier plants lived in the sea like modern seaweeds, absorbing water and carbon dioxide directly through their surface, not needing internal tissue to transport liquids from specialized organs like roots. We can also obtain other sections by the film pull technique, which show that Cooksonia was a land plant. We can see the plant's cuticle, with stomata, pores, inside those little dark rings which are the guard cells. Gases pass through these pores during photosynthesis and respiration. They're openings in the waxy cuticle which protects the plant from loss of moisture. Aquatic plants wouldn't have cuticles. Now, modern green plants have leaves. Cooksonia didn't have them. If we look at one kind of plant that's still alive today, a club moss, we can trace certain fossil evidence of the stages by which its leaves, at least, may have evolved. Here's another Silurian plant fossil, Sordonia, a few million years younger than Cooksonia. It's a larger plant. If we look at it closely, we see spines projecting from its stem. These can be investigated using another paleobotanical technique. The rock containing the Sordonia is broken up, then placed in a plastic vessel to which hydrofluoric acid is added. This is a highly dangerous substance and must be handled in a fume cupboard using protective gloves and a face mask. Over a period of time, the hydrofluoric acid dissolves away the rock minerals, leaving the organic matter of the fossil unattacked. This organic residue is then filtered off. The fragments of Sordonia are washed thoroughly, then examined under the microscope. You can see the little spines. Expert examination shows that they do not contain any conducting tissue, any veins, so they're not really like leaves. Here's another way in which they can be examined. They can be coated with silver, then examined under the scanning electron microscope. Here's the picture coming up. There are 3 of the spines. It's possible that their function was to increase the plant's photosynthetic surface area. Here's an electron micrograph taken using the machine. They're not leaves, but they may represent a stage towards true leaves. Now let's look at another primitive Devonian plant, Asteroxylon, long since extinct. This is a cross-section of its stem, fossilized in chert. And here's yet another important technique used by paleobotanists. The rock surface is this time etched with hydrofluoric acid. It takes a little time, after which the fossil is left standing slightly proud of the rock surface. A thorough rinsing with water, and now she uses the cellulose acetate peel technique. The surface is dried, acetone is poured on, then a thin film of cellophane is pressed down onto the surface. When this is pulled off, it brings with it a very thin section of the fossil. This is mounted on a microscope slide. A cross-section of the stem of Asteroxylon, clearly showing the star-shaped xylem. Now let's jump forward to our modern club moss, a Lycopodium, to see how it's related to the long-extinct Asteroxylon. A cross-section of its stem, mounted and suitably stained, looks like this. Again, with star-shaped xylem, suggesting that it evolved from some such primitive plant as Asteroxylon. Returning to Asteroxylon, here's a longitudinal section of its stem. What we're looking at is the stem, of course, with the xylem running down the middle, and there are the traces of conducting tissue running out to the bases of the spines, but not entering them. Now, in the present-day club mosses, the leaves have conducting tissue extending right to their tips, like this. Asteroxylon may not be the ancestor of the modern club mosses, but it does show one of the stages by which they may have evolved from some similar plant. Here's a fossilized transverse section of Asteroxylon again. Those dark smudges surrounding the outline of the stem are actually sections of the spines. The outline has, of course, been deformed in the process of fossilization. We may suppose that, simplifying a little for clarity, it looked like this. By examining many transverse sections taken using the peel technique, it's been possible to reconstruct what Asteroxylon must have looked like when it flourished in Devonian times over 400 million years ago. Once again, we're not saying that this is the ancestor of modern club mosses. The yellow sporangia and the small spines represent simply an earlier level of organisation compared with our Lycopodium today, with its simple leaves and its sporangia. So there were some, only some, examples of the value of collecting fossils and studying them in order to understand plant evolution. We saw Cooksonia, probably the earliest land plant. No leaves, just a photosynthetic stem. And here's a slightly later plant with more branching, but still no leaves. Sordonia, with spines which may represent an increase in photosynthetic surface, but they're still not leaves. Asteroxylon, with its rudimentary leaves, presenting us with one stage in the evolution of our present-day club mosses. Now for another fossil series involving an invasion of the land. The American Museum of Natural History in New York, like all such major research centres, owns an enormous collection of fossil material. We've picked several important specimens which give us some idea how the first four-legged animals, the first tetrapods, may have evolved. First, there were the fish, purely water-dwelling animals. It's likely that tetrapod limbs arose as modifications of fins. In this fossil specimen, the fin bears no resemblance to the limb of a tetrapod, although there is a shoulder girdle here. But there are fossil specimens from what are called lobe-fin fish from Devonian times. Here's one in the position the lobe fin occupied. There are the radial bones of the fin, but here we see a humerus, a radius, and an ulna, homologous to the three bones in a tetrapod forelimb. And there's the shoulder girdle. This is Sauropterus, a famous fossil lobe fin, the humerus, radius, and ulna, and the shoulder girdle. There are the radial bones. Compare this with the forelimb of an early amphibian tetrapod. Again, the humerus, the radius, and the ulna. And now the bony rays have become the hand and finger bones of the limb. The girdle. There's a ball and socket joint with the head of the humerus. In the living animal, the girdle was in this position. Dr. John Maisie, curator of fossil fishes at the museum, talks about Sauropterus and the evolution of tetrapods.
The fossil of Sauropterus is historically very important because it was collected over 150 years ago and clearly shows a pectoral fin with elements in it the same as the main bones in a tetrapod limb. And for a long time it was regarded as an example of an ancestor of tetrapods. Now, in real terms, it cannot be an ancestor because at the age of the rocks from which it comes, it's already too late because there were already early tetrapods in existence. So while we can't make any definite statements about ancestry, we can at least see some homologies between the pectoral fin of these fishes and the tetrapod limb, which suggests to us ways in which the tetrapod limb may have evolved.
The first tetrapods were amphibians. Here in the Department of Zoology at Newcastle University in England, a research student is working on the fossil remains of one early amphibian, Jenny Agnew.
This is a specimen of an animal called Pholidopetton scutigerum. and it was so named by Thomas Henry Huxley, who was a great colleague of Darwin. It was found in 1868 down a coal mine, and it was found in the roof of a coal mine, and the records say that it was taken out with great difficulty. It was then placed in the collections of the Bradford Philosophical Society, where it was embedded in plaster and placed in the Cartwright Hall Museum at Bradford. I'm in fact the first person to restudy this animal since 1926.
She first of all cleans away parts of the matrix surrounding the fossil using a dental mallet. Then further cleaning up by blasting with a jet of sodium bicarbonate particles. Each bit of the fossil has to be cleaned up as much as possible. Here's the animal's jawbone. The various sections are photographed. From each photograph, a tracing is made. The fossilized bones are broken and displaced, and Jenny Agnew has to work out how each part of the skeleton must have looked originally. Dozens of drawings are produced. They can be used as templates so that with dental wax, flat replicas can be made up, then bent into shape to produce a three-dimensional model. Here's the side of the skull. She also uses another technique. From her sketches, here's the roof of the skull, she makes clay models of what the original bone structure is likely to have been. From these three-dimensional reconstructions, it's possible to gain a good idea of what the animal looked like. What's the point of all this difficult and time-consuming work?
This animal lived 300 million years ago, and one can only be fascinated and curious about what life was like in those days. The other reason for studying these particular beasts, which may seem rather arcane to anyone who doesn't know anything about them, is that this particular group of animals was for a long time, and in fact I think by some still are, considered to be related to the ancestry of reptiles. This is no longer held to be so by a lot of people, but it's only by constructing three-dimensional and two-dimensional pictures of them that we can say whether or not this is the case.
In the next film, we shall start by looking at the fossil evidence for the emergence of the reptiles.